will be recorded for educational and promotional purposes. My name is Ruchi Shah, Scientific Project Manager at ILSI Europe, with a background and experience in leading food ingredients R&D and bioprocessing for 4 years. I joined ILSI Europe in 2024. I proudly lead ILSI Europe's Horizon Europe project involvements and food safety task forces, namely risk analysis for food safety and microplastics initiative. Today, we have an excellent panel of presenters, including Neil Buck from General Mills, who will be moderating and helping us on the scientific discussions of this webinar, Georgia Porcaro from University of Liège, Juan Carlos Correa from Shell, and Carlos Martin Elberga from Cargill. This webinar is open for questions and discussions after each speaker presentations. We highly encourage, if you have questions, please leave them on the chat so efficiently address them during the time frame allotted for the Q&As. ILSI Europe's mission is science-based guidances in the food sector to foster collaboration between academia, industry, and government bodies to facilitate consensus building. We bring together with experts with different point of view and stakes and interests and opinions and build consensus based on science and the evidence available. ILSI Europe is a European branch of global federation, leading the federation with the biggest number of members. Our portfolio involves food allergens, food packaging, food-related contaminants, microbiome, new approaches in food safety, nutrition development, and healthy aging, including European Commission-funded projects. Scientific integrity principles apply to all of ILSI Europe's activities, including inclusivity and transparency. As a non-profit organization, ILSI Europe operates through in-kind contributions of experts and relies on financial support mainly for corporations and institutions. The design, review, and execution of ILSI Europe's scientific research proposals and activities are independent from the funding sources. ILSI Europe's scientific research proposals are selected for their relevance to public health. All activities are designed and implemented in such a way that highest quality standards are achieved. Expert groups are unbiased. They consider the totality of evidence and incorporate different opinions and points of view that interpret according to the grade and the consort of the norms. How we work is we identify challenges and key emerging issues through a horizon scanning. We develop a task force. This task force develops a proposal that goes forward to develop an expert group where 50% are compulsorily involved from the academic institutions, followed by the industry and some public authorities. We publish publications on guidelines, reports, and we disseminate and then create an impact from them. We publish impactful science-based guidances. We disseminate our deliverables very efficiently. From here on, Neil Buck will be helping me to moderate this discussion. He is a corporate toxicologist from General Mills. Following postgraduate study of toxicology and a PhD on food additive safety, Neil has spent 25 years within the industry, currently as corporate toxicologist with General Mills Inc. The safety support Neil provides is split between product innovation, emerging safety concerns, and regulation thereof. He is active in various stakeholder fora in both the US and the EU on topics relevant to continuously improving consumer protection, including ingredient safety, contaminants, and allergy management. He is proud and reputed member of ILSI Europe in multiple initiatives such as food contaminants and risk analysis for food safety task forces. I give the floor to you, Neil.
Yeah, thank you very much, Richie. So I won't take up too much of your time, but just to provide a brief introduction before we get to the main event. I have the honour of being the current chair of the Food Contaminants Task Force in ILSI. This is my declaration of interest. So essentially, I'm an employee, full-time employee of General Mills. I don't have any other declarations of interest. So, Yeah, if we move to the next slide, please, Richie.
Yes,
Yeah,
of
so
course.
thank you. What I'll do is very briefly talk about a new activity in ILSI, so the Risk Analysis for Food Safety Task Force, just to raise awareness and also as a bit of an advert in case there's anybody who may be interested in particular what we're doing in that new task force. And then I'll talk about the Food Contaminants Task Force, which is where to date we've managed the topic of mineral oils from an ILSI perspective. So next slide, please. So the Risk Analysis for Food Safety Task Force, as mentioned, this is a new task force. So risk analysis really describes the overall process of risk perception, risk assessment, risk communication, risk management. And what we're aiming to do in this task force is understand the interplay between those different disciplines, so the inputs and the outputs and how decisions are made along that continuum. So what we're going to do is understand how those interrelationships have developed, either by design or organically, understand those inputs and outputs and the trade-offs that happen, and hopefully identify some opportunities where there is a potential for improvement in the overall risk analysis process in terms of either better informing or even protecting consumers. So we have a new activity that just recently started, and that activity is looking at case studies of food safety incidents. And the reason why we're looking at those case studies is because what we identified is that those food safety incidents that happen over a defined period of time are really a window into which we can understand, evaluate what's happened in terms of that overall risk analysis process, you know, the risk perception, the assessment, communication, and the regulatory decision making. So to understand how that's worked, particularly in the EU. So we'll be looking at mineral oil as one of the topics in terms of previous food safety incidents, so the market action that has resulted from the risk analysis that's happened in the past, and also previous incidents such as what happened a few years ago with ethylene oxide, or even earlier with the veterinary drug fipronil in eggs, and the market action that happened there, and the decision making and the assessment, etc. So anybody that's interested in finding out more about the Risk Analysis for Food Safety Task Force, please reach out to Ruchi. The email address is there. So it is a new activity. So there's an opportunity to perhaps get involved and help to shape how it moves forward. So, and then in the next slide, I'll talk about the Food Contaminants Task Force. So the Food Contaminants Task Force has been operating for many years now and has addressed lots of different topics in the contaminant space, which I'll talk about in a second. And really what we aim to do is to consolidate multi-stakeholder knowledge on what do we understand about a food contaminant, what don't we understand, where are the gaps, and also we also produce best practice guidance on what's the multi-stakeholder position in terms of how a particular topic might be managed, whether that's analytics or whether it's mitigation, etc. So we have a number of ongoing activities that are starting to come to a head now. So we had an activity running on risk benefit assessment. So how is that deployed so far and how could it be deployed? We have an activity on prioritization of natural toxins. So which mycotoxins are of predominant concern from a public health perspective and which can have the best opportunities for mitigation. And we also have an activity that recently started on incident management. Now, this activity is to derive a best practice on how do you do rapid risk assessment. So what's the technical risk assessment process rapidly to respond to an incident based on the data that you have available. And the Food Contaminants Task Force also run a rolling series of seminars, so early career scientist seminars, which have been increasingly successful over the years. So the next one is in October in Prague. So if you're interested in that, this is all managed by the colleague in ILSI, Conrad. His email address is there, and there's also further information on the website. So the sorts of activities, if we go to the next slide, that Food Contaminants Task Force has been involved in really falls into three buckets. There's like risk management activities. So we recently did a publication on what do we really mean by contaminant? What's the definition of contaminant? And identified actually there isn't a great deal of clarity on what is meant by contaminant. As I mentioned, we produce technical guidance, such as, for example, in the analytics space or the mitigation space. And then, of course, we review emerging issues, such as the multi-stakeholder workshop that we previously ran in the mineral oil space, which led to a publication in 2021 on a that we term the gaps and roadmap. So if you go to the next slide, please, Ruchi. So this is the publication, and really this is the preamble for the webinar today. So what has changed since this publication, since this workshop? So what's happened since then? Because actually there were a lot of gaps that were identified previously in terms of analytics, the lack of a standardized validated analytical method, interferences that can happen in that method, the problems with the availability of more detailed characterization of individual congeners within a mineral oil hump, lack of data on exposure, lack of understanding of the actual toxicological concern related to a lot of congeners that may be present within mineral oil. So lots of gaps there. So what has happened? What's changed since 2021? Where are we now? And just my last slide before we get into the main event is just to mention that there is a kind of elephant in the room in that we do have pending regulation. So there is draft regulatory text, which is in development. I think it's now on its 7th iteration of development. The European Commission is hoping to finalise this draft text soon between the member states, and the proposed applications for the enforcement would be from the start of 2027. So what we're looking towards is having maximum levels for MOA, so aromatic hydrocarbons, and monitoring and aspirational targets or indicative levels in the case of MOSH, so the saturated hydrocarbons. So that was my little introduction. We get to the main event now. So we move on to Georgia. So Georgia is going to talk to us about the latest happenings in the analytical space. And Georgia is extremely well placed to give that discussion for us. So Georgia is a full professor of analytical chemistry at the University of Liège, at the Glenblue Agrobiotech department, and Georgia, unless I misremember, has been working in the field of MOA, MOSH, mineral oil analysis for at least 15 years, and is really an expert in the development of methods for both sample prep and also these hyphenated chromatographic methods, etc. So over to you, Georgia.
Thank you so much for the introduction, Neil. Let me share my screen. Voila. You should see the presentation now, right?
Yes, we do.
Perfect. So thank you for the introduction. And yes, as Neil introduced, I will talk mainly about the advancement on the analytical side. Well, here there is my declaration of interest. So I'm an employee at the University of Liège, and now I have a project going on mineral oil funded by the Belgium research funding. So what happened since 2021? So the publication that Neil mentioned. In that publication, there was a nice summary of the different aspects, analytics, exposure assessment, hazard characterization, and risk assessment, giving a state of the art back then in 2021, and the current gaps that were present in that period. And on the analytical side, which is the focus of this presentation, the summary was done in a table that I reported here, so systematically highlighting the different gaps and proposing action required to fill this gap. So let's try to go through the main one and what has been done over the last 4 years to fill this gap. Of course, at the first point, there was the fact that there were no official method with the sensitivity required. The only one was the EN 16/1995 that had a limit of quantification of 10 ppm, which was way too high compared to what already back then was started to be asked by the food retailers. In this regards, in 2024, an update of this method, the ISO 20122, was published with the scope of covering vegetable oils. And what it was done from the analytical viewpoint was to add a saponification step before the following purification and final determination. And this addition allowed to reach a limit of 3 ppm for the MOSH and 2 ppm for the MOA fraction. Other important gaps that were highlighted are the lack of an harmonized or validated sample preparation protocols that cover a wider range of matrices, the epoxidation, which is part of the purification step in the sample preparation workflow, but also the difficulty in the integration of the results, on the interpretation of the results, and the lack of convention on how to report these results. And all these gaps concur to a big problem in this field, which is the high uncertainty of the results that are produced by different laboratories around the world. And as you can see in these images here, what we obtain as a response from the instrument is a rather complicated chromatogram that needs to be interpreted. So how these gaps were covered? First, to try to cover the difficulty in interpreting these results, the Joint Research Center has published two guidance to support the analysts on interpreting the results and the chromatogram, to decide what to include and not to include in the quantification of the mineral oil, but also in how to report the results. So a big difference between the two versions of this guidance was that in 2019 was requested to report the quantity of mineral oil based and subdivided in carbon fraction. This in 2023 was removed, and only the total amount of mineral oil between C10 and C50 is now required. And a more detailed explanation on how to calculate and to report the LOD and LOQ was provided in this guidance. So to try to reduce this uncertainty, as well as detailed reports on how to report the results, where it specify specifying all the different steps that had been performed in the sample prep, to show a chromatogram, how to describe the chromatogram, and so on. So on these regards, big advancement has been done. In the same guidance, the JRC, the Joint Research Center of the European Union, also tried to tackle the problem linked to the harmonization and validation of the sample preparation. And in particular, a decision tree is there reported. And let's have a focus on this decision tree. You can see that there is a big chunk of this decision tree that pertain to the sample preparation steps for this determination, and is basically steering the analyst in when to apply or not to apply determined sample preparation step. And on the bottom part of the decision tree, there are all the information regarding data interpretation and data integration. Nevertheless, despite this guidance, the data interpretation and data integration account for more than 20% of uncertainty. This is an estimation that has been done with particularly critical matrices. So what can we do to reduce the uncertainty that is caused by these three main steps: the sample preparation, the data interpretation, and the data integration? So for sure, we need to improve the sample preparation, and still it needs to be done, increase the knowledge and the understanding of the interferences, standardize the methods and the procedure, and harmonize them between different labs. Again, regarding this data interpretation and data integration, we have support from the scientific literature, where several chromatograms and how to interpret them are reported, the guidance itself, the new ISO method also reports several examples on how to interpret the results and how to integrate the results. The JRC also organized a very nice exercise to support the data integration here to extreme on how this interpretation can be performed and how different is the results. As you can see, we have almost an order of magnitude of difference if the integration and the interpretation is not performed consistently. And he reports how this should be done, explaining in details how. Also on the part of the sample preparation, so to try to standardize the sample preparation and to reduce the uncertainty, many efforts have been done over the last 4 years, and the JRC has been the main driving force of this, and organized several interlaboratory comparison and study on different matrices to try to better understand and to harmonize the procedure. So you can see here muesli, pure mineral oil, edible oils, and again, a big exercise on the infant formula. Nevertheless, despite this effort on harmonizing the entire workflow, we still have problem because the different methods were not consistent between them. And in particular, in all the new methods, the saponification step has been incorporated to reduce the sensitivity, so to reach the sensitivity that is required by the upcoming regulation. And it has been highlighted and it's been reported, which is, I think, is the most serious thing, that still a big uncertainty is present. These are the results of an example of an olive oil from the proficiency test performed by JRC. You can see that even within the same lab, there is a big uncertainty in the results. In this case, not a detailed discussion on the internal standard was performed, but if we move to the ISO method and the infant formula, both of them applying saponification, there was a big discussion on the internal standard that are used for quantification. Indeed, in the procedure, we add internal standards to control that everything goes as it should, and the ratio between these internal standards should be 1. But in the different methods, we have a discrepancy, and the acceptance of this discrepancy is different if you look at different documents. So as you can see here, ISO accept up to 1.25 of discrepancy. The infant formula report that a 13% of discrepancy is observed between the internal standards, but even worse, if we look at the JRC guidance published in 2023, they there assume basically that a discrepancy up to 1.15 can be accepted, should be avoided, but is a kind of intrinsically there. So this discrepancy can again generate uncertainty, and it can be a problem. Of course, all the documents say that the goal should be to have 1. But in a way, it opened the doors to the presence of uncertainty, which is always there. So what we did on our side was to study more in detail what happened on the saponification step. And we define a change in the proportion of the solvent used in this step to try to have a more consistent recovery of the internal standard, which allow for a more consistent quantification of the mineral oil extracted from the food sample. In this regard here, you can see how the distribution of the different solvent was changed comparing the ISO method to the new procedure that we develop. If we look at the results without the metrics, you can see that here is what is expected. In the ISO method, the discrepancy is rather big. And of course, if we add the metrics, the discrepancy is even higher for the ISO method. We have discrepancy with the new method as well, but it at least is reduced. Although it's still very dependent on the matrix that is analyzed. And this is still under study to understand which are the main reasons that lead to this discrepancy and how can we keep it under control and tune the sample preparation when needed. If we look at the sample preparation, so we go back to the decision tree, so to the workflow, there is also another very critical point that generates still a lot of uncertainty in the results, and is the epoxidation, that is the purification that is used in the MOA fraction to remove the natural occurring interference. So sometimes we can have chromatogram like this, where the mineral oil, the MOA fraction, is completely hindered by the presence of these natural occurring interferences, so we are unable to quantify them. So the routine method suggests the use of an epoxidation to remove these interferences. But the problem of this method is that it actually also react with the mineral oil, the MOA in particular, that we want to quantify. And it depends on the matrix and the particular condition applied, because it's a matter of kinetics. So it has been estimated that we can have a loss of 20–40% of the MOA in this procedure, which is a problem. Many protocol study has been done to try to improve the epoxidation, to improve the purification on one side, and the recovery of the MOA, so to reduce the impact on the MOA fraction. Nevertheless, we haven't succeeded yet with the epoxidation to improve it at a point that is reliable enough. But other alternatives are under study, like, for instance, this work that has been published early this year, where we studied the selectivity of the ILSI column itself. So using exactly the same column used to separate the motion MOA in the official method, changing the gradient that was used in the ILSI, we are able to eliminate most of the interferences. Here you have example of palm, sunflower, and olive oil with different kind of natural occurring interference that are there. And applying the new method, we were able to completely remove the carotenoids and squalene derivatives, although still failing with some of the interference present in sunflower, which are different terpenes. But the good thing is that with this method, we have a recovery all over the different edible oils, over 90%, which is significantly higher than the recovery obtained with epoxidation. And for this method, we have been asked to present it as an official method, so to submit to an interlaboratory trials to make it official because of the improvement compared to epoxidation. The other advantage of this method is that if we subfractionate the purified fraction, we can characterize separately the 1–2 aromatic rings from the 3 and plus aromatic rings that have a different toxicological relevance, as Juan Carlos will explain later on. Moving on the gaps and the filling of it, one big gap that was highlighted in 2021 was the lack of a validated confirmatory method. GCMS was also proposed back then, but over these years, GCGC FIDMS has emerged as the confirmatory method to use. And this was also recognized by the EFSA opinion. So while in 2012, it timidly mentioned GCGC as a possibility, but say that it was not routinely applied. In 2023, when the update was published, GCGC took a main part in the analytical procedure. It was recognized that we can also lower the detection limits, meaning that has a more important role also from a quantification viewpoint. And indeed, the advantages of using GCGC is evident. If we look at a simple chromatogram in a 1D-GC, we just have a hump. If we do a GCGC, we have a full characterization of the MOA in terms of number of aromatic rings in this direction and degree of alkylation in this direction. If we look at the MOSH fraction, we can also separate the MOSH from the MOSH analog, for instance, the POSH in this chromatogram. But also it can help to have a more accurate quantification, like, for instance, where to put the baseline, which is a problem in the uncertainty linked to the integration, because in GCGC is chromatographically separated. Also with some interferences here, for instance, we have an example of a samples where the internal standard are completely covered by the interferences. In GCGC, we separate them chromatographically, and so we can quantify the hump that is down here without any problem. Of course, it's not the panacea. We still have some problem. These interferences that I showed also before from sunflower oil are still there. So we still need improvement in the sample preparation. So in this regards, also big advancement have been done for the quantification and to validate the quantification in GCGC. Although a guidance on GCGC how to interpret the results from a qualitative viewpoint is under development and should be published by the end of the year, while several RIN tests are ongoing to validate the quantification side of GCGC. Also, the last gap is the missing of reference standard. As far as I know, there are many projects going on right now at the European level to develop a reference material. So with this, to summarize, there are much more support to interpret the results nowadays. There's a validation going on for the GC/GC use. There is a creation of reference material ongoing, but still we need an improvement on the sample preparation, in particular the epoxidation step. And we should harmonize better what to do with the internal standard. And so still the sample preparation remain kind of critical. With this, thank you for your attention, and I'm open to answer your questions.
Thank you, Georgia. Wonderful update. It seems that a great deal has happened over the past few years, thanks in no small part to yourself and colleagues. But so we are open to questions. We have 2 questions in the chat, both from Elton Go. So I'll read them out. So some labs provide results as total integrated and some states total calculated. What is the correct reporting result?
So previously it was the calculated because it was asked to do the carbon fraction, but now it's asked to report the total motion more, so it should be the total integrated one.
Right.
But it depends if then there are some company that still ask for the fractions, as far as I know.
Right. So the second question is, how can I improve baseline stability and eliminate false peaks in blank run in LCGC FID?
This is a very good question. I think that all the analysts are fighting with this. So to improve baseline stability, there are some tricks from a pure chromatographic viewpoint, that is to, for instance, have a temperature ramp going down when you cool down your system. It may help, but the main problem is that it depends on the samples that are injected mainly, which can, if they are very heavy, they can kind of build up in the system and so cause fluctuation of the baseline. So there's not much that we can do in this regard. If not running blank constantly to be sure to go back to a good baseline. For the other peaks present in the blank, this is, of course, a matter of having the purest solvent possible. What we do also in our lab is often to distillate our solvent to try to purify even more and to try to work as clean as possible. So everything things contribute to these extra peaks. But the single peaks are trimmed during quantification and so should not be a big problem.
Gotcha. Yeah, understood. Thank you for the clarity. There are obviously fundamental weaknesses with the FID method. So we have another question from Angeliki Stavropoulou. So she says, in a recent conference, it was mentioned that it is not possible to separate fully MOA from interfering compounds, even with GC/GC. Is some work ongoing for improving that aspect of the new method?
Yeah, so as I show, there are some compounds that can be separated chromatographically, but not all. Some remain on top of the GC/GC part of the chromatogram that we are interested in. What is ongoing is actually on the purification on the LC side. So before going to the GC/GC.
Right. Understood. Thank you. And we have another question. Is there an online solution for LC purification as a replacement of epoxidation, or it's only offline for the moment?
For the moment, it's only offline, but we are working to try to make it online as well, as we are working on improving the purification in there.
Great. And we have a question that's just come in from Stylianos Agriropoulos. Excuse my pronunciation. In the everyday lab report we receive as industry, how easy is it to have POSH and MOSH differentiation? Thanks.
So it depends if is a 1D analysis, so an LCGC analysis is more difficult, but again, it depends because you can spot the pattern of the POSH on top of the MOSH, but then it depends on the relative proportion of one of the other, if this can be quantified separately. In GCGC is easier, so it depends which kind of instrument is used in the lab.
Gotcha. And Elton Goes come back and said, based on your experience, what are the preliminary steps to prevent carryover? Is there a cleaning method?
Depends carryover where, if is referring to the GC column or the LC or the transfer line, because there are different strategies there. So maybe I can answer to this question offline with specification on the question.
That's great. Thank you, Georgia. I'm just checking the time here. We do have a minute or two left. If there's no further questions coming in from the attendees. Do you think there is a time in the future, near future or far future, where the FID method will no longer be used? So the hump integration method and the predominant method will be GCGC, because it does seem that a large number of samples will potentially have interferences. And so ultimately GCGC is the only reasonable approach to understanding, what's there from a mineral oil perspective.
So from a discussion with people that are really performing routine, so that are doing a lot of samples, they still say that is a minority of sample that has problem of interferences.
Okay.
So is a big chunk of the production. Like for instance, edible oil, most of them have problems. So in the field of edible oil, possibly at 90% need a GCGC. So in that case, it can be surely the way to go without doing a GC FID.
Okay. And you mentioned that there is an interlab study that is being started on 2DGC.
Yeah.
What's the timeline associated with that, and to get into the point of having an accepted standardized method?
I'm not sure about it. I'm participating, but I'm not coordinating it. So I don't have the perfect timeline, but from the experience on the ISO method, it will require at least a couple of years.
Okay, understood. Thank you. So that was wonderful. Thank you, Georgia.
Thank you.
So we've got to the end of the allocated time for questions in perfect timing, actually. So we move on to the next speaker, which is Juan Carlos Carrillo. So as it says on the slide here, Juan Carlos is a senior toxicologist at Shell. So Juan Carlos over many years has led efforts to evaluate the safety of mineral oils, and has been involved, in fact, in many cases chaired a number of stakeholder fora, including a mineral oil hydrocarbon speciality task force, which is resulting in a number of key publications, which are important references to understand the safety aspects of mineral oils. So looking forward to the update on the toxicology side. Thank you, Juan Carlos.
Thank you for the kind introduction. And thank you for the invitation to give you an update on what has happened after the last ILSI workshop. I hope you can see my screen. First of all, let me give you my declaration of interest. Obviously, I'm interested in mineral oil, but I guess that's not what they were asking me to.
Can I stop you, Kwan, for a second?
Yes?
We just see your presentation not in the presentation mode. So maybe it's the different screen that you are on.
Yeah, okay. Sorry.
No problem. We are still in time, don't pray.
Let me start this. So I was saying I am a full-time employee of Shell. And obviously, we are manufacturing mineral oils and also synthetic oils. So I guess that's the...
Can I stop you, Kwan? I really cannot see the screen.
No, I will share it now.
Okay, great. Okay.
Yeah. Can you see it?
Wonderful. Thank you so much.
Yes. some remarks from our lawyers, but I guess I can throw this to the side and give you an update of what has been happening. So, what was the status in 2019 regarding MOSH? At that time, we were still calling MOSH as a catch-all term for all types of alkanes that were present in the fraction. Then, also at that time, we knew that the Fischer rat, which is was the predominantly model for the assessment of oils and waxes, had a higher retention of MOSH in the tissues. And in particular, the endpoint of interest here was the granuloma difference between humans and F344. So the F344 rat develops after so-called MOSH exposure epithelioid granulomas in the liver, whereas humans, they do show hydrocarbon retention, but these are non-adverse, definitely not the same type as the Fischer rat develops. And while in the rats, they look very distinct, and also chemically, if you analyze them chemically, they will be different. In humans, you see mostly oil droplets, and you don't see the type of strong reactions. So this reaction to the presence of MOSH was to some extent suspected to be related to the chemical structure. So the F344 showed the strongest granuloma response to waxes, and we know that waxes consist predominantly of N-alkanes, whereas the lipogranuloma in humans related to the accumulation of naphthenics. So the cycloalkanes present in the MOSH fraction. And in human livers, there were no N-alkanes in their biopsies. There was already out there a mode of action proposed by Adenuga, which was based on a MOSH assessment. So here, David did not distinguish between oils and waxes. He approached this from a MOSH perspective, so really not making any distinction between the different types of alkanes that are present in the fraction or the type of responses that you see in humans and rats. There was some indication that there was a higher absorption rate for MOSH in rats, that they had some kind of low metabolization, and that they were more sensitive to an inflammatory response. And that was the status back then. Since then, we published a paper, and this is an interesting one for different reasons. We tried to write it in a way that even non-toxicologists will understand it. So it's open access, so I invite you to go through the almost 80 pages that it was, that it was in its form when it was submitted. So it's a double column and 2 pages, so probably it's only 30 pages or so. It's paper published in Critical Reviews of Toxicology, and that's important because this journal allows you to really do a deep dive into what was really going on. So it is a puzzle, and I think we managed to solve it to a great extent, at least to come to some overview of the regulatory developments and how we got there. So it was important to review everything that had happened and come to a differentiation of MOSH according to alkane subclasses, and that includes N-alkanes, iso, and naphthenics, and to come with a biological explanation of this type of alkane accumulation and the relationship between the biological effects and the structural elements of the chemistries involved. And based on all these puzzles that came together, pieces of the puzzle, we were able to update the mode of action based on chemical specificity. So how does this mode of action look like? So we are using an adverse outcome pathway. This is a tool that toxicologists used in order to put together all the pieces of the puzzle and come with a very illustrative way of explaining what's going on. The first thing that you do is that you look at the stressor agent, and in this case is the N-alkanes. We know that one of the first key events in the Fisher rat, it is inherent decreased metabolism. So it is a rat that not only doesn't metabolize the N-alkanes very well, but also the other types of paraffins. Then that decreased metabolism leads to a dose and time-dependent accumulation. And in particular, the N-alkanes start to accumulate. And because of their accumulation, then we go into the second key event, which is their crystallization in the liver. After they crystallize, you see the typical reaction of the appearance of a foreign body that triggers the activation of the Kupffer cells present, which then lead to an increased phagocytic activity with a positive feedback loop that recruits even more. And at the end, the third key event is the formation of the epithelioid granuloma, which is not seen in humans. So this is a very typical F344 reaction only to N-alkanes that are present. And this is really an effort to the rat to clear the crystals, the N-alkane crystals. And for the F344, this is adverse. But as I said, this is not relevant for humans. So that was taken into consideration in the 2023 EFSA assessment, where the panel concluded that the formation of the epithelioid granuloma leading to liver inflammation in the F344 rats exposed to MOSH is not a critical endpoint relevant to humans. So that means that all the previous assessments that use the F344 endpoint for risk assessment are no longer valid. That was then further taken out in the wax safety assessment, the food contact material 93, in the 2022 opinion of the SEP panel, where they again used the AOP-based F344 liver effects as not being relevant. The European Wax Federation presented a new study and analytical data, and now there is no concern for hydrocarbon accumulation when exposed to food contact material 93, so the hard paraffin wax, with a new NOAEL of 9 grams per kilogram body weight per day based on the SD rat, instead of the old NOAEL, no observed adverse effect level of 20 milligrams per body weight per day. So this is no longer valid, and therefore now there is a new SML proposed of 5 milligrams per kilogram. So that's new. So what is relevant to humans? And this is a study we did where we compared GTL synthetic paraffin to mineral oil. And this is a very straightforward study design. You take 2 groups of rats, you feed them either mineral oil or gas to liquid, so a synthetic Fischer-Tropsch oil. You expose them for 90 days. After that, you switch the diet, you give them clean diet, you let them rest, and then collect their organs. And this is really to explore the differences, the chemical differences between the mineral oil typical MOSH components and a synthetic oil, the GTL oil. And as you see here from the chromatogram, the line of the N-alkanes gives you, let's say, the orientation. So the GTL is above that N-alkane line, very linear little branch type of compounds, whereas in the mineral oil, you see the cloud underneath the naphthenic compounds, which you see in this graph, how that affects the retention in the liver. So clearly the one above is the MOSH, and the one in red below is the GTL. So the line at 90 days is when they get the time to rest. So clearly both are eliminated, but the mineral oil is much slowly eliminated compared to the GTL. And this is in SD rats, not in the F344, that is even slower to metabolize. So this is a rat that is used now for the assessment of MOSH according to EFSA. What we have here is really the residues at the end of recovery. So on the top, you see what was fed to the rats, and below you see what was found at the end. And what you see in the last set of chromatograms, where the cycloalkanes are analyze, you clearly see that it's the cycloalkanes that are heavily retained compared to basically no indication of cycloalkanes in the GTL, because the GTL does not have that type of constituent. It is isoparaffinic, whereas the mineral oil is a mixture of isoparaffins and cycloparaffins. And that was published, and that data was submitted to EFSA. And in their opinion of 2023, they recommend the further investigation of the structural features of MOSH to allow a better comparison of mineral oils with GTL oils. So you see here that MOSH has to be chemically differentiated, and that GTL is not just MOSH, it's something different, much more quickly metabolized, because structurally it is different. What is now going on with MOA? Where we left it in 2019, again, MOA was a catch-all term. There was no dose response for MOA for hazard characterization. And there were questions about the 1 and 2 ring MOA. So what happens if they're long or short alkyl chains in the molecules, in the 1 and 2 ring molecules? This is the work from Dan Lay. I see that she's present here in the seminar, so you can ask all the questions to her. But it was, I think, her work that brought more light into what's going on when you add different types of alkyl chains to the ring systems. In essence, when you add a very long alkyl chain, typically the ones that you find in waxes or in oils, the metabolism will be performed on the side chain and not on the ring system that will bioactivate them and make them carcinogenic or mutagenic. So you clearly see that there is a very big role of alkylation, and by and large, the type of MOAs, 1 and 2 ring MOAs that you find in refined oils and waxes are of the highly alkylated type, and that has to do with manufacturing conditions and also boiling points. And these papers really give you an indication that it does play a role where the alkylation is found and the length of it. But for the refined oils that normally go into food contact materials, the type of MOA that you find present is 1 and 2 rings of highly alkylated, where the oxidation will happen at the side chain. We also published some differences between polycyclic aromatic compounds and the concept of MOA to understand what is really relevant for hazard assessment and eventually for risk assessment. And here in the paper, we describe what are the typical levels of MOA as you go through the refinement process, how the MOA levels vary, but also the response in mutagenicity tests, for example, and what will be the corresponding IP346, which is the indicator of whether a material has been properly refined or not. So that's also interesting information that has come to the pile of papers probably you have next to your desk and you still haven't read. So for the CEP panel, there was also an indication of MOA assessment. That was the wax assessment I mentioned before, where there is the new study, the 90-day study that also included indication of 1 and 2 ring MOAs. And the panel concluded that there is no concern on the 1 and 2 ring MOAs present, and there is no concern on the current level of pH levels, pH, because the manufacturing conditions are such that the pHs are extremely low because of the type of requirements that are around the quality that has to be guaranteed on the food contact material 93. So I'm working at the moment on a risk assessment on MOA types. So what I'm doing now is I will look at the risk assessment. So it's not only hazard assessment, but here we are also considering the exposure on the 1 and 2 rings. And here you're going to approach this from a NOAA point of view because of the properties of these MOAs that are present that are not carcinogenic, mutagenic, but you still have to come with some kind of guidance on the levels that will be considered safe. Then I'm also doing that for the three-ring MOA on a TTC approach, because here you would have to distinguish where the alkylation takes place, whether it's in different positions, and also the database for the type of phenanthrenes is not conclusive. And then also on the margin of exposure approach for the three-ring and more. And with that, the summary here is that the liver effects in the F344 are no longer relevant for risk assessment. So that's why we have a set of new recommendations for the waxes, the food contact material 93. There is also for at least the oils, there needs to be a chemical distinction between the naphthenics, which are of low toxicity but has a potential to accumulate, versus the GTL, which is rapidly metabolized because they are made of isoparaffins. And the MOA also needs further distinction, 1 and 2 rings, 3 rings, and more than 3 rings. At least from the toxicological point of view, this is important. And with that, I'm happy to take your questions. And maybe I have the answer.
Well, thank you so much for that comprehensive summary, Juan Carlos. So the floor is open to questions, as Juan Carlos mentioned. Do we have any questions related to the toxicology? Okay, whilst we're waiting, I have one. So EFSA, in their last opinion, discussed that there is an inadequacy of data on the 1 and 2 ring congeners. What, in your opinion, is the data that is missing and is needed?
Yes, I have been revising data that is available on certain refinery fractions. And normally here, you would think that you will need data from MOA present in what you're finding in food, but you also can take a worst case scenario and try to apply data on aromatics that are 1 and 2 rings, that are not carcinogenic, not mutagenic, but they have repeated those data that are further up in the refinement process. So what I'm trying to do is to take that as a starting point and use it as a worst case. And then if there is no problem with those type of aromatics, as you increase the carbon chain length, then basically the longer you are from in the alkylation degree, the safer the molecule becomes, because it's less bioavailable, and also the side chains make it even more difficult to become hazardous. So that's the approach I'm taking for the 1 and 2 rings.
Gotcha. Okay, thank you very much. That's clear. Yeah, and if I may ask another question, actually,
yes, please.
Is a pretty fundamental one. Is that when we were first dealing with this topic of concern of mineral oil aromatic hydrocarbons potentially contaminating the foodstuff, there was concern that there was a misunderstanding that actually any 3+ ring congeners are unlikely to be present in the food chain, at least from mineral oil, as they are not present in refined oils. And so the assumption that 3+ ring congeners are present in under a MOA hump, and therefore the regulatory action that was taken was perhaps not in line with the tenets of the precautionary principle in terms of the amount of precaution that was being applied, because it was difficult to see where those 3+ ring congeners were coming from. I don't know if you have any comment around that.
Yes.
On well, yep. Okay.
I do. In fact, the approach here is to have a generic approach, because, as you rightly say, if you look at the refined products, what you find there is 1 and 2 rings. However, not always, as I understand, when you analyze food, the contamination comes from legal mineral oil. So it can also come from processes where there is a contamination that is not necessarily coming from an refined oil.
Mm-hmm.
So here is really trying to look at this from a much broader perspective. So you know that you have a food contact material that is refined and all that. So you know what type of MOAs are present, and you should know how to deal with it. But if you have an unknown source, then you should also be able to deal with that. So that's, yeah. And therefore, it's very important here to emphasize that when you're running your analysis, you should distinguish between the ring types, because only then you know what kind of risk assessment tools or what kind of data set you can apply to what that particular set of MOAs that you're finding.
Mm-hmm. Understood.
Yeah, so that's why I'm presenting this from different buckets. So how do you deal with this type of contaminations?
Thank you so much. And we do have a question in the chat from Dingvu Gayan. Is the MOA chemical composition described in the TDS in a way that the end user can make its risk assessment?
In the TDS, technical data sheet.
I suspect so, yes. So the information that travels along with the
and here is important. That's a very good question, because normally the term aromatics has to be in context, right? So when you have a refined product, and then you look into the TDS, for example, and you see the type of aromatics that are present, that will be, if it's a refined oil, it will be the 1 and 2. Yeah. And then that's the information that I think somehow is missed in translation, because the terminology MOA in itself does not exist in technical specification from petroleum products.
Right.
Yeah. So the MOA terminology is actually a food terminology.
Mm-hmm.
Yeah. But if you're looking into the food contact materials, you talk about aromatics, you talk about pHs, but then the term MOA has to be taken into the context, into the language of the petroleum products, to make a clear distinction of what is it what you're talking about.
Understood. So there is potentially something that could be done there in terms of transparency of
well, at least to understand what's being talked about. For example, if I look at the current FDA requirements, so there is a requirement there for UV analysis. So when you're measuring by UV, you would be measuring according to the wavelength that you're required to measure, and that will give you also the type of aromatics that you're assessing. But that is not called an FDA MOA test. So I think what we need is to have some kind of a translation tool to understand what is it what we say are the aromatics that we are measuring, and what does that mean in terms of, let's say, MOA language. I don't know if I explain myself.
Well, I understood. It was I thought it was clear. Thank you, Juan Carlos.
So it's already that, but it needs to be translated.
Got you. So we do have one last question, but we are approaching the time a lot. Oh, sorry, Richard.
Can I stop you, Neil? There is one more question right before the one we just connected. So it's from Dario Maggioni, and he doesn't have his microphone, and he apologized. And he would like to ask if currently there is any maximum limit for MOA and or MOSH in food or food contact materials in the EU or the USA.
Again, okay, MOA, let's put it into the context. There are maximum limits of aromatics that are allowed, and that is either for a technical grade white oil, for example, that will be an H1 oil. So there are already FDA levels, so they are measured by UV light, and it can be done like that. So you don't need to run GC times GC. And there is also the pharmacopoeia tests that determine the level of aromatics that you can have present. What there is not at the moment is in mineral oil an UV method that determines the total level of aromatics. So that will be the 1 and 2 rings plus the 3 and more. The current pharmacopoeia test for food contact material 95 is based on the hazardous, the 3 to 7, but not on the 1 and 2. Mm-hmm. So there are, but you need to be able to translate them into more. And I think that's where we have to be careful that whatever legislation that comes doesn't undermine the pharmacopoeia, the FDA test, and the same European levels simply because you get lost in translation.
Mm-hmm. Yeah, thank you so much for the clear explanations, Juan Carlos. Conscious that we are starting to cut into Carlos's time, there is a question that's in the chat from Barbara Moretti, and perhaps we can answer that online afterwards. You can take a note of that, Ricci, and deal with that afterwards to make sure that we stay on time. We should move
Indeed.
to the presentation of Carlos Matan Alberca. So Carlos Matan Alberca is senior scientist at Cargill. So is really an expert in the area of lipid analytical, predominantly for contaminants, which is an area where he's focused on for the past 5 years or so, and really has a broad experience in a range of analytical methods and bringing those to bear in terms of method development on analysis for contaminants, specifically in edible oils. So Carlos has also been a part of various multi-stakeholder projects in this space, and there's some important publications that he's been a part of. And so we're hoping for Carlos to bring us home here in terms of the latest understanding of mitigation or remediation, specifically in edible oils. So thank you, Carlos.
Thank you. Let me share my screen. Do you see my screen now? Yes, and the full presentation, the presentation mode. Perfect.
Yes.
Okay, so first things first, the declaration of interest. As you see, I am full-time employed by Cargill, and the rest of my time I fully dedicated to my wife, my daughter, and my hobbies. So no other conflicts or interests. So good afternoon, everyone. So my name is Carlos, and yeah, I work for Cargill, as Neil has said, as a senior scientist for contaminants and lipid analytical topics. So I started working on this topic almost 5 years ago. So I got that picture, that we have been speaking about in this last two talks of the state of the art of this topic.
Carlos, I'm just going to stop you for a second. I think your
Yes, sure.
microphone is making weird noises, and we hear a lot of.
Do you hear me now better?
Yes, we do. Perfect.
Yeah, two colleagues stopped next to me and they were chatting.
Thank you.
Sorry for that. So as I was saying, when I started working on this topic, on the MOSMOA topic, that was 4 or 5 years ago. So I could get that picture, that state of the art. And since then, yeah, I've been comparing day to day with the current state of play. So that's what I want to explain to you today, what we have been doing these last 5 years in Cargill. So at Cargill, we have taken a science-based comprehensive approach to understanding and managing mineral oil hydrocarbons across the entire value chain, from fundamental research and source identification to exploring alternative supply chains, refining optimization, and advanced analytics. We have left no stone unturned, and we have translated this knowledge into actions, implementing every known and feasible control and remediation measure. And our goal has been always very simple, very clear, to do the right thing, so for food safety, for our customers, and for consumers. So today I'm going to share with you a summary of actions we have been taking to prevent and remediate mineral oil hydrocarbons in edible oils and fats. So just briefly speaking to you about Cargill, it's a family-owned company with over 150,000 employees in 70 different countries, committed to nurturing the world in a safe, responsible, and sustainable way. So in the edible oils family, we operate in 18 different countries, and we work with up to 16 different types of oils, including canola, coconut, sunflower, palm, soybean oils, etc. So this global footprint gives us access to a wide range of raw materials and full visibility across the supply chain. This is a unique position that has allowed us to study mineral oil hydrocarbons in a deep and also implement solutions at every stage, always with food safety as our top priority. So preventing mineral oil contamination is absolutely critical, and the prevention starts with understanding. That's why we have conducted a thorough investigation of all known and potential sources from plantations and mills at origin, but also throughout the whole transport, storage, and processing stages. We have looked into every corner, and not just to eliminate the obvious risk, but also to uncover hidden or unexpected sources. So for example, in APAC, our colleagues conducted a study at one of our production sites to assess whether the construction of a nearby highway could influence in the most small levels in raw materials. And this is a clear example of other factors, no, like the environmental factors that can also play a role in this type of contamination. We need to think beyond the factory walls. We need to think beyond the supply chains. These contaminants might come from many other sources as well. Another case that came from our own laboratories is where, we discovered that the sampling jars where we were using were leaching compounds into the oil samples. So basically artificially inflating the MOS values. So these learnings have been, as I said, incorporated or included in our new way of working. One of the most important lessons that we have learned is that continuous monitoring is essential to prevent mineral oil contamination. So that's why we have implemented a robust control system that spans the entire supply chains to ensure full control and minimize risk, from origin through transport and into our refineries. We also purchase large volumes of crude oils and other raw materials, and we need to ensure that every batch meets our safety standards. So this means not only controlling our internal processes, but also maintaining strict oversight of what we receive from suppliers. As you can see in this graph, since 2020, we have been investing over 1 million euros annually in external analytical service. This allowed us to monitor raw materials and final products with precision and consistency. We all need to keep in mind that this level of investment is not feasible for every company or every region or across the globe. But for us, it was and is still a necessary step to ensure food safety. As part of our broader strategy to manage mineral oil hydrocarbons, we have dedicated thousands of hours to understanding how refining can help reduce or eliminate these contaminants. Our teams have run extensive simulations, tested a wide range of variables and conditions, and even developed and evaluated new technologies. From this work, we have drawn some clear conclusions. Mineral oil hydrocarbon compounds with carbon chains below C25 can be fully removed using conventional refining techniques. And this is very good news. In the C25 to C35 range, removal is only partial, and for the above C35, conventional refining has limited effect. And these findings are consistent with those from an independent series of studies led by Professor Porcaro, by the way. Their work focused on the removal of mineral oil hydrocarbons, aromatic hydrocarbons, under generic refining conditions. They found that MOA compounds below C25 can be significantly reduced, including both the 1 to 2 ring MOA and also the 3+ ring MOA. However, for the C25 to C50 range, particularly the long chain alkylated 1 ring MOA, the refining effect they concluded is minimal. So it's quite important, no, that both studies agree on one critical point, that the most toxic fractions can be effectively removed. So the non-alkylated and short chain alkylated MOA and the 3+ ring structures can be removed, no, just by conventional refining procedures. And this is a critical outcome. But we need to do something else. We need for an effective manage of these contaminants. We recognize it early that having a strong in-house analytical capabilities would be essential. So that's why we have made substantial investments, not only in the state-of-the-art equipment, but also in developing the skills and expertise of our colleagues around the globe. Today, multiple Cargill locations across all regions are equipped with the standardized LC GCFID method, and this methodology is widely used for routine lab control, production control and monitoring, and also for product release. And this is a very big effort, no, that our food safety colleagues has done, no, in our company worldwide. In parallel, in our R&D teams, we have built advanced capabilities using LCGC times CC TOF-MS FID. This approach allowed us to go far beyond routine testing, has allowed us to run deep root cause analysis and also support innovation, and has helped us to better understand the behavior of these contaminants in different matrices. And this journey has made us stronger internally, but also has allowed us to contribute meaningfully to the global scientific community on this topic, sharing openly what we know about the analysis of mineral hydrocarbons with analytical colleagues. So I want to wrap up my presentation with this case study. This case study illustrates how source knowledge and analytical precision and understanding come together, and it's also an example that speaks about those gaps that Georgia has mentioned at the beginning of her presentation, and also build up on top of what Juan Carlos has said about pH versus MOA components. So in this study, we investigated how traditional drying methods used in the coconut copra production might influence mineral oil hydrocarbon contamination, or more precisely, how they might interfere with mineral oil analysis. One of the methods we examined is the so-called smoking method, where the husked coconuts cut in half are dry over a direct heat source in contact with a smoke, as you can see in that picture. We manually extracted the oil from these pieces of copra and analyzed the crude oil, coconut oil, avoiding any kind of cross-contamination source. What we observed was a distinct hump in the MOA region, of the chromatogram, as you can see in that picture, in that chromatogram. However, this was not due to MOA itself. It was caused by PAH compounds, the polycyclic aromatic hydrocarbons, which are generated during this smoking process. And as Juan Carlos said, and also the EFSA scientific opinion said, PAH can interfere with the MOA analysis, and this is a piece of evidence for that, and that can lead to false positives. With the implementation of the GC times GC TOF-MS method, we gained the ability to precisely identify the nature of complex humps in MOA chromatograms. In this case, we analyzed again, a couple of years after, another crude coconut oil sample. First, we studied it with standard ILSI FID method, and the MOA value that appeared there was something like less than 2.5 ppm. But the chromatogram clearly suggested a high presence of PAH compounds. So thanks to the GC times GC based method and a thorough study and additional confirmatory analysis, we were able to confirm that this signal was almost entirely due to the presence of PAH compounds with virtually no MOA presence. So again, this distinction is critical. PAH and MOA are different type of contaminants, and PAH we know that can be effectively fully removed during the refining process. So as a result, this oil, for instance, after refining, will be fully, how do you say that? Meeting the legal limits for both PAH and MOA, and it will be safe for consumption as well. No? So this case highlights a key point, especially now that authorities are going to consider future legal limits for MOS and MOA. Setting the maximum limits on crude oils, where interferences are more common and harder to interpret, increases the risk of false positives, and especially when using the standard LCGC FID method. In my view, maximum limits should be applied on refined oils, where the analytical signal is clear, where the product is also closer to what the consumers are going to finally consume. So it's critical also to note that the GC times GC TOF-MS method is not widely available. And as Jorge has also mentioned, we are still far from its standardization. It's even more expensive, the use of GC times GC, than the standard methodology. And it's also way more time consuming, it's technically very demanding, requires super specialized analytical specialists running the equipment, and it's not feasible for routine in many regions across the globe, at least today. And this makes even more important that the regulatory frameworks are fully aligned with analytical realities. So coming to the end of my presentation, conclusions. So we have shown a clear commitment to prevent this type of contamination. To avoid it, we need to combine multiple strategies. There is not a magic formula, unfortunately. Collaborative and independent studies have shown potential for remediation or removal of these components using just current and standard refining capabilities. And very important that the most toxic fractions can be removed. And having access to cutting-edge analytical technologies is also key to give support to R&D initiatives, innovation, root cause analysis, etc. So thank you for your attention.
Thank you very much, Carlos. We still are in time, and we are open for questions. I still don't see questions on the chat.
I may have one.
Please go ahead.
Carlos, so you indicated that the C30 plus fraction is what you have difficulties with by removing from your
Yes.
processes. Yeah, and that is, do you know the chemical components there? Is it naphthenics or is it isoparaffins? Do you know?
We see that it's difficult to identify these components. We are still trying to investigate what remains there because it seems to be slightly different to what comes in. So it's still a very big, difficult question to answer, Juan Carlos. But if we look at the LCGC FID chromatograms, we see that that part of the C fraction is still there. It doesn't disappear as it happens with the rest, with the below C35. But with the GC times GC, we should be able, no, at some point, GC times GC, TOFMS, we should be able at some point to identify those components and try to understand what is happening there or why we are not able to fully remove them.
Which is interesting because from a toxicological point of view, these are exactly the same type of components that tend to be retained the hardest in the tissues. So what we see is that lower viscosity oils have, let's say, mush components that are more easily metabolized and eliminated from the body. When we talk about naphthenics and highly isoparaffinic components. And as you go up with the molecular weight and the viscosity of the oil, then you start seeing the retention effect, which then has a peak around C35 because afterwards they are no longer bioavailable. So then that plays a role there.
Okay, I'm taking a good note of that, Juan Carlos.
Thank you. I don't see more questions for Carlos, but I would like to go back to Juan Carlos. One of the questions that was left unanswered in the previous time slot for your Q&A, maybe we can go back to Barbara Moretti's question where she wanted to ask, with the differences in toxicological assessment based on different aromatic rings, the GCGC analysis, it seems in the future, the best solution for the MOA contamination evaluation. What do you think?
Well, yes, it is not the panacea, as George says, but it's an important element in a decision-making tree process. That's how I come to understand this. So I guess that there are different tiers that probably would need to be implemented here. And when you have absolutely no idea about the origin of the MOA, that's when the GC times GC is helpful. And that's basically what Carlos just showed. I found his example like a typical case study that you run an analysis, you see high levels in MOA, and then you think that MOA is MOA. And by running the GC times GC, you see that MOA is not always MOA. It can also be pHs. So from our community of those interested in this issue, I think we have to come with some terminology and also clarity when we are talking about aromatics, when we are talking about MOA, what exactly do we mean? Because then you can apply the right tools to address it.
Well, thank you. I hope it was answered, Barbara. Yes, we still have 1 or 2 minutes left.
Yeah, maybe Neil, I would like to dare to answer to the question you have made to Georgia, if I may.
Yes, of course, but Neil
is not anymore there. Okay.
No, he had to go for a personal emergency. But
so he asked it a question about the future of the standard method by LCGC FID and GC times GC, and Georgia gave her answer right. So I would like to build up on that answer. How I see the future on this for this methodology or for the MOA methodology. So once we know everything about interferences, once Georgia finalized this wonderful piece of work that she's doing now with the removal of interferences through LC separation instead of epoxidation, Then what we should do is to take a different direction. Instead of making the analysis more and more complex, we should try to simplify the analysis. And we should try to make it useful and available widely, for all type of QC laboratories across the globe. So every company can afford this. Every entry level user can run this type of analysis. So that should be the aim. That should be the future, how it looks like for the Mosmo analytics. So we need to start simplifying at some point, because the analysis is so expensive, so complex, so difficult to manage or run. So we need to know, as an R&D, I have that strong R&D mindset, right? We should start working as soon as possible towards that simplification of the Mosmo analytics for the future.
Thank you, Carlos. Georgia, do you have some inputs on this comment?
I agree with Carlos. Although I'm a lover of GC and I would like to see GC everywhere, and using a routine method because I think it's a fantastic technique. Of course, the goal is to have a full picture with complex sample of some complex instruments, but the ultimate goal is to simplify, to use these tools to simplify the life of everyone. So then have a very target analytical method for
Excellent. Thank you. I think it is a very incredibly wrapped up comment to really end the webinar in the nice note that we need to work on simplification. And before I close the meeting, thank you very much for all of you, especially the speakers and Neil Bach at the same moment for moderating. The alignment and the content of the webinar being so useful and insightful. We will be posting the recordings of the webinar online at ILSI websites, ILSI YouTube channel, and information on the LinkedIn as well. For any questions, if you want to get in touch with the speakers, please send an email to me and I will put forward all of you to the speakers directly through me. Thank you very much for all of you and your time, and have a good evening.
Thank you.
Thank you. Goodbye.
Bye-bye.